The goal of my talk today is give you a genomic perspective on SARS-CoV-2, which is a causative agent for COVID, where the virus has been or as it's going. As a bit of a background, I lead the Temple Viral Evolution Group, which is an interdisciplinary computational biology group. The primary focus of what we do is methodology and tool development and a lot of collaborations. So I'm actually an applied mathematician by training. That was a long time ago, but I'm a bioinformatician and computational biologist. The reason we were able to plug into SARS-CoV-2 research is twofold. My lab has been doing translational research on viruses for close to two decades. The primary focus of our earlier research was on HIV-1, which unfortunately remains a major problem. Actually, SARS-CoV-2 exacerbated a lot of the issues in developing countries in terms of treatment. Suppression of HIV-1, influenza A, hepatitis C, and now, as I'm sure you're aware, everybody in science who has any connection to SARS-CoV-2 is working on SARS-CoV-2 by necessity. So we're doing that as well. What my group is mostly known for is by developing state-of-the-art statistical and machine learning techniques and software for studying the action of selection, natural selection on viruses and other organisms. And we're a part of the Institute for Genomics and Evolutionary Medicine, which is an organization that Mike Klein helped establish. One of the mission goals is to study disease dynamics. So that's what we're doing. Just a couple of screenshots. This is the primary software package that we developed called Hi-Fi. It's been in development for about 20 years, gets on the order of 500 or 600 citations per year, pretty well used. That's a great way to sort of, get Temple name and reputation out because, it's all linked, the research and people cite it. One of the things that we also do is allow people to run their data through a web server that we maintain on CSD infrastructure. So we have a server room on the 7th floor of the building. And this is actually where we get about 80% of our users. This is very easy. You don't need to download anything, just click a few buttons. One of the interesting things that we found is that we keep track of what type of data people analyze. So our server typically processes about 7,000 to 10,000 jobs per week. And these are fairly computationally intensive jobs. And we started tracking what fraction of them had coronavirus data. And you could see, so week one is sort of week one of the epidemic. As we go through, the orange bars here indicate that people upload coronavirus data. So by our estimates, about 10% of all the jobs submitted to our server since March 2020 included coronavirus data. So you can view this as sort of a bit of a public service that we do to the greater research community because they can analyze their data for free. The use is picking up again now with the nucleates, and we're sort of expanding capacity to allow people to analyze more data. So you can have lots of perspectives on SARS-CoV-2. Typically, I would begin my talk by telling you why this matters. What's the biomedical application? Obviously, I don't have to do that now. SARS-CoV-2 is front and center in our lives. It's been front and center of our lives for too long, in my opinion. Unfortunately, it'll probably remain there for a while. You can view SARS-CoV-2 as a virus, as a patient that's infected, whatever it is that you envision. This is what I see. This is the viral genome. This is about 30,000 letters there. It's all there. This is the reference genome. I pasted out of GenBank. So this is what the substrate of what our lab operates on. So just lots and lots of viral sequences like this. It's hard to believe, but this is sort of the entire composition of the viral genome that's caused us so much grief. And as far as viruses go, it's actually about 3 times as long as an average RNA virus. So it's sort of as long as RNA viruses gets, about 30,000 nucleotides. So why does it matter? One of the interesting aspects of this pandemic, which you may or may not have been aware of, is that this is what I would call a real-time genomic generation pandemic. So if you look at the chart on the top right, I borrowed it from this organization called GISAID. GISAID is an abbreviation, I believe, for Global Initiative of Sharing All of Influenza Data. So it's an organization based out of Germany that's been sort of created with the goal originally to allow public health officials and research organizations to share surveillance data on influenza, like about 20 years ago. Now they've taken over as the primary repository of data, genomic data that's being generated by researchers worldwide on SARS-CoV-2. And you can see this, the shape of the curve at which sequences are being generated is your classical exponential growth. So, you know, in January, there were in the order of 350 sequences, genomic sequences of the virus. As of yesterday, there were 415,000, right? And this rate is not slowing down. So people are actually accelerating generation of the data. So that is unique. That has never happened before. Previous pandemics occurred when we didn't have the sequencing capacity. So this is something that we should be able to exploit. Obviously, having a lot of data creates opportunities, but it also creates challenges. The opportunities are we can learn a lot of things that were impossible to do before: early detection of novel strains, tracking viral evolution, trying to predict where it's going to go. But the challenges are technical because in our field, we never had to deal with this much data. So all the typical things with data sharing, data distribution, data analysis, data presentation had to be overcome. What our group does is study patterns of genetic variation, so mutation or combination in viral genomes. And we use statistical methods and machine learning to basically understand where this virus came from, what it's doing now, and attempt, and by attempt, I really do mean attempt. This is an open problem that has never been solved in the history of viral evolution, but we can try to do better this time to predict future evolutionary trajectories of the virus. So just to give you a perspective, I'm sure you've all heard about the new lineages. Lineage is just another word of a variant. It's just a branch, an offshoot in an evolutionary tree that arose in the United Kingdom, South Africa, and now Brazil. And there's this a key mutation which occurs in the spike protein. It involves position 501. It changes one residue in amino acid N to another residue in amino acid Y. And this is how you can actually obviously see what's happening here. This is a collection of sequences, a small part of the viral genome. So each row is one particular sequence. You can see there's some sequences from South Africa that were isolated in December. There's some references, sequences from England, from Japan, from Ukraine, and so on. And if you scan down this column, the 501 column, you see that all the South African sequences have a Y and all the reference sequences have an N. So this was an amino acid change in the protein, which turned out to actually have a significant functional impact. It made the virus more transmissible. In South Africa, you had another change. You had this E2K change at position 484. And this actually has to do a lot with how effective antibodies are at binding and neutralizing the virus. So these two mutations combined, it's been estimated to make the virus about 13 times more transmissible, right? So one mutation is 2.3 times, and combined, they're about 13 times more transmissible. And recently there was a study that came out of South Africa that we contributed to that said that having these mutations actually makes it much harder for people that became infected with an earlier viral variant to neutralize this type of variant. So now imagine having 400,000, a very, very large table of these letters, where we're basically looking at patterns of variation and trying to understand which ones of them matter and which ones of them don't. So for instance, if you look at the first column, there's a blip of N here, but it's kind of rare and it probably doesn't matter as much where you have these dramatic shifts. So viruses, especially RNA viruses, evolve. That's what they do. They always have, they always will. This is nothing new. And SARS coronavirus is no exception. So there were two, I'll just give you a couple of descriptive facts. So it's been estimated that SARS-CoV-2 makes about 8 genomic changes per 10,000 nucleotides per year. Because there are about 30,000 nucleotides in the genome, that means that an average virus over the year, right, so it's transmitting through multiple individuals, is going to accumulate about 24 differences. Sounds like a lot. And it is. It's about 100,000 times faster compared to humans, but it actually is slower than other viruses. So it's about 10 times slower compared to influenza, HIV, and hepatitis C. Part of the reason is because SARS-CoV-2, unusual for an RNA virus, has an enzyme that makes fixes some of the errors that it makes copying itself. So early on in the epidemic, there was a paper published by several prominent virologists that had titles like this: "We shouldn't worry when a virus mutates during disease outbreaks." That is correct. That's what they do. And then in September, the coronavirus is mutating, and that's fine. So far, I would maintain that it's still fine. Discount what the press tells you. The press has historically been using every single mutation in the virus to make people, in my opinion, unnecessarily scared. So I'll try to give you, some more background there. So let's actually, coronaviruses are ancient. In fact, in 2013, while I was still in San Diego, we collaborated with researchers in Hong Kong to try to understand when coronaviruses emerged as a species. So some of the key findings we made is that they have an evolutionary history much longer than previously estimated. In fact, coronaviruses as a viral species are probably 300 million years old. And they've been infecting birds and mammals, or basically the entire history of birds and mammals. So what this means, when you have such a long-term relationship, that has allowed some animal species, for example, bats, to evolve strategies to coexist with coronaviruses and vice versa. So they infect bats, bats don't seem to care too much, and then coronaviruses use bats as a springboard to jump off into other species. But these types of estimates you can get by comparing genomic sequences from viruses isolated from different hosts. SARS-CoV-2 is not the first coronavirus to infect humans and become endemic. In fact, there were 6 different coronaviruses previously identified in humans. You had 4 endemic strains. They're basically common cold viruses. So you had 2 discovered in the 1960s, this OC43 and 22E. So alphas and betas are different types of coronaviruses. You have alphas, betas, gammas and deltas. You have 2 reported in early 2000s, and you had 2 epidemic strains, of course, one SARS-CoV-1, which had a brief outbreak in 2002 and 2003, and MERS in 2012, had a very small, thankfully, outbreak because it had a 35% fatality rate, but it was actually very poor at transmission. So viruses, because they have to infect humans and jump species and evade host immune responses and develop drug resistance, they're subject to very strong selective forces, which drive rapid changes in their genomes. Viruses and some of the bacteria, what's called measurably evolving pathogens. You can actually watch evolution take place because the generation cycle is so small, and you can, within a week or 2 weeks in HIV in particular, you can see mutations arise. But viruses also are not free to change. Their genomes are subject to constraints on structure. They have to be able to maintain the molecular structure and function. They have to be able to infect cells via receptor binding. They have to be able to replicate. So some changes are not allowed to them. From my perspective, if you have 400,000 genomes, you can study patterns of change in their genomes to identify, which sites are actually subject or might be subject to adaptive evolution and which sites are constrained. So we can find and classify those genomes. So looking at sort of the history of SARS-CoV-2, one of the things that, people aren't that interested in it anymore, but early on it was a big drive to try to understand where it came from. Our best guess at the moment, we don't have the definitive answer, but the best guess is horseshoe bats, where it came from. So a canonical tool for this type of analysis would be something called a phylogenetic tree, which you see here on the right. All the dots are individual viral sequences. On the x-axis, you have sort of an estimated time, chronological time. So, going all the way back to the 1200s, to the, dark ages. So SC2 is SARS-CoV-2. SC1 is SARS-CoV-1. So you see they're actually quite different. They're, probably most recently shared their common ancestor in the 1400s. So right around the time of the New World discovery, I suppose. And the most recent isolate we have for bats is from about, we're guessing that the most recent common ancestor of SARS-CoV-2 and the bat virus that we have occurred in the 1980s. So these types of analyses allow us to determine closest animal relative to SARS-CoV-2, try to describe the sequence of evolutionary steps which give rise to the early SARS-CoV-2 genomes. And one of the questions that we asked is whether or not the virus had to fine-tune its genome during the early stages of spread in humans. So there's this paper that's now exists as a preprint that we did together with our colleagues in the UK. at the University of Glasgow Center for Viral Research. It's going to appear in PLOS Biology this week. The title of the paper is Natural Selection in the Evolution of SARS-CoV-2 in Bats, but not humans, created a highly capable human pathogen. I'll come to the figure in a second. I'll just give you the sort of the breakdown of the major findings. We looked at great detail at what the virus was doing in the first 9 months of the pandemic, so up to October of 2020. And we found that the virus was actually not doing very much at all. It was not showing much evidence for selective pressure. And we think this is because it didn't have to. It's because most humans never seen a virus like this, so there is no selective pressure. And evolution never does anything it doesn't have to do. So if there's no selective pressure and the virus is able to transmit as it is, it's going to do that. It's going to accumulate some changes, but none of them are consequential. Of sort of greater concern, and we've known this, and especially coronavirus researchers have known this way before SARS-CoV-2, is that coronaviruses are what one would call a family of generalist viruses, which means that they're actually quite good at switching hosts easily and rapidly. There's a history of these viruses going from bat species to bat species, jumping off to things like pangolins and snakes. And that seems to happen very, very easily. It did not, and once it emerged from the bat, this particular virus, it did not require many additional changes to spread among humans. This is actually somewhat unusual. If you look at the history of some of the other viruses, for example, Hong Kong flu, right, the 1968 H3N2 outbreak, that virus got into the human population and was pretty infectious, but it required some fine tuning. So you could see in the first year, of course, sampling in the 1968 era was much shallower, some change. And here we basically don't see anything. Now, as areas of concern that we find is a future emergence or recombination event of other coronaviruses is likely, which means that something like a SARS-CoV-3 virus could be sufficiently divergent to evade either natural vaccine acquired immunity. And we see this with SARS-CoV-1 and SARS-CoV-2, because if you were infected with SARS-CoV-1 back in 2002, 2003, this would not protect you against infection with SARS-CoV-2. It's sufficiently different. So we advise that in addition to dealing with the pandemic that we have now, we should not forget that there is always another animal reservoir there that a new virus can come from. So we should dramatically ramp up surveillance in the human-animal interface. SARS-CoV-2, SARS and COVID-19 causing viruses are here for the long term, unfortunately. And the picture sort of shows our best understanding of where the virus came from. So it's, there's a wide reservoir of viruses in bats. They transmit from bats to other bats. Changes in evolutionary environment force the virus to make changes, like switch to different bat species. Then, and this is the part where we don't actually know what happened. We don't really have the intermediate steps. So we have sequences from pangolin. There might be an intermediate host where the virus came from, but we haven't sampled it yet. The best information we have now is that what we got as a SARS-CoV-2 came from directly from a bat transmission. And that bat virus arose as sort of a combination of other coronaviruses infecting large colonies of bats, probably in Southeast Asia. So moving on, which mutations matter, right? So most mutations in the viral genome, as I asserted to you, are neutral, so are ignorable, but some are very consequential. It's like this 501 mutation. One of the things that we've been doing over the last 9 months is we developed a SARS-CoV-2 genome selection dashboard. It's a publicly open website, which you can go and look at. We analyzed every single publicly available genome sequence at least weekly. In order to do this, we had to invest some time to overcome a number of technical challenges in scaling up our tools to handle about 50 times more data than the original design specification. We aim to identify which among the thousands of mutations that you will see in these 400,000 genomes, pretty much every single mutation you could imagine is there, just because the virus mutates a lot. About 80% of positions in the genome have some variation, but most of them don't matter. We pride ourselves on developing open source tools, allows other scientists to use them, and we develop interactive web-based exploration to allow you to do that. So what makes a mutation important? We use a couple of categories here. We use our tools to predict whether or not it's subject to positive natural selection, meaning whether or not there is selective pressure for the virus to acquire a variant there. We also look for variants that simply increase in frequency over time. We look for variants that are located in the region of the genomes that are targeted by known or hypothesized immune responses. We look for mutations that are in some sense unexpected. So something we could not have predicted based on the historical evolutionary patterns of coronaviruses. And our goal here is to continuously monitor the viral genome for evidence of adaptation. So things like escape, fitness change, et cetera. And here's a screenshot. Don't expect you to look at all the details. I'll just highlight a couple of things here. So this is the evolutionary trajectory of this N501 mutation. If you look at the time series here in the bottom right, you'll have the calendar time on the x-axis. Different colors represent different amino acids at this position. So this light yellow is N. You know, in the beginning, it was exclusively N. Then sometime in July, you started picking up some variation there. And then sometime in October, you can see that this variant, the Y variant, really took off, right? So it increases in frequency. We also have a way to visualize where in the world it occurs. So it's very prevalent. For example, it's a UK variant, so it's common in the UK, but it's also fairly common in South Africa. It's found in Asia, Japan, Australia, New Zealand, and so on. This variant is also subject to immune selection and so on. So these, and you can look at every single site in the genome using our tools to see what it's doing. Now, we've also collaborated with, this is a busy slide, I'll talk you through it. But 2 weeks ago, this preprint came up from South Africa entitled Emergence and Rapid Spread of a New Severe Acute Respiratory Syndrome Related Coronavirus, lineage with multiple spike mutations in South Africa. We contributed to the study. It's been accepted to Nature, so it's coming out sometime this week, I think, or next week. The key point that we observed is that following a period of neutral evolution that our previous study showed up to October 2020, something happened in 2020 November. So here's a sort of a time series plot where each row is a particular location in the genome in the spike protein, which is a functionally important part of the protein. If there's a bubble there at a particular, this is the y-axis. The x-axis is a time series. So this is when we did the analysis. So for example, if you look at site S614, the one that has a low across, that was a site that was probably the first that people found as potentially selected. But we found it pretty much throughout the entire series of our analysis. So we picked it up in March and it stayed turned on until present time. Now you can see there's a dramatic shift that happens in November. There are just a couple of blips of something showing up as selected up to this point, but then in November, you have about 20 sites in the genome lighting up all at once. You couple this with the fact that we now have 3 independent emergences of this N501 lineage in the UK, South Africa, and Brazil, and they're completely evolutionarily independent. They're not the spread of the same strain. They were developed separately, separate evolutionary events. So we hypothesized that this occurred because the virus is now subject to selective forces. We don't know why. We're actually actively working right now with a team of researchers to try to come up with some hypotheses. One of the hypotheses that's an obvious one is that the virus is no longer encountering just a naive individuals, immunologically naive individuals. Some of the individuals have been infected with another strain. Perhaps they have partially protective immunity. So a new virus gets in, it's now actually being pressured by that immune response. There's also complicated dynamics because South Africa and Brazil have large fractions of population that are infected with untreated HIV and so on. If you're interested, you know, I'll be happy to discuss at the end. One of the other things you can ask is, could we have predicted where changes would take place? So we can predict some, but not all. So these are the 3 different lineages: the UK, that's also designated as B117, South African, and Brazilian. These individual numbers like N3 or 5 or 852 and so on, these are specific sites in the genome that were diagnostic for this lineage. They're different. I mean, you can see they all have 501, which is a canonical one. The red line is basically when they started emerging. So the question is, did any of our analyses pick up some signal at those sites before, which would indicate bubbles occurring to the left of the red line? And you can see we can pick up a couple sporadically. but we miss a number, right? So we couldn't have predicted 501, we could have predicted N3, but not 501, not 570. We can do better for other clades. So Brazilian clade would have actually been pretty well predicted, but we need to do better. So we can, our tools are somewhat predictive, but they're not, an oracle of what the virus is going to do. And if anybody tells you they can tell you what the virus is going to do, don't believe them because they can't. That has never been done. Now, to sort of look at the more positive side, so I've shown you that the virus is making a lot of changes in response to immune pressure. We can also look at parts of the genome that are not making any changes. So nearly all nucleotide sites will change because of random mutation, but some will have little or no variation in amino acids. So this is a hallmark of so-called negative selection. The virus simply cannot afford to change amino acids at these positions. It may try, but those viruses don't survive, so we don't see them. These sites could be attractive targets for vaccines that the virus cannot easily circumvent, or drug targets. For example, and its good news is there seem to be a lot of sites under negative selection, including over 150 in the spike protein, which is immunogenically important. So here on the right, I showed a little ribbon plot of this blue part is the receptor binding domain of spike. ACE2 is the human protein that it binds to infect the cells. This is the interface where they bind. And the orange spots are the sites that the virus seems to not be able to change, right? So those are the sites where we've seen no variation. The variation that we see indicates that the virus is not capable of easily acquiring mutations there. So, including some in the area that's next to the binding motif. So looking forward, SARS-CoV-2 is here to stay, unfortunately, and will continue to evolve. All RNA viruses have, and they will continue to do this. Our tools can serve as a part of what we call the early warning genomic system, with a goal to, for example, identify new sites in the genome where the virus may be evolving in response to selective forces. On the flip side of this, we might be able to keep track of constrained sites as possible vaccine or drug candidates, and to continue surveillance of related species for possible spillover candidates. To give you an idea, this is a screenshot from our yesterday's analysis. There are 11 sites that came up as newly showing signatures of selection since January the 11th. This little chart is a cartoon representation of the viral genome. Individual proteins are indicated here, and the red slashes are where these positions occur. If you zoom in on this one, site 766 in NSP3, for example, and if you look at where it's doing anything, it seems to be increasing in frequency in the United States recently. So there's this little blip here. It's subject to immune selection, and it has some variants that we could not have predicted based on viral evolution in bats. So this is a potentially interesting site that we could look at further. I've given you a brief overview of what we're doing. One of the benefits of being a computational biologist is that you have a lot of collaborations. Some of the things that we're doing, we're actually involved in a large-scale interpretive study with researchers in the UK and South Africa to try to figure out why these recent lineages occurred. In collaboration with Dr. Kumar, who's the director of our institute here at Temple, we've developed and published as a preprint, it's being reviewed now, a new approach to easily classify and track viral lineages. An interesting twist about this is what repurposed cancer genomic tools for SARS-CoV-2. I'm also working with teams at Penn State and Johns Hopkins to look at how the virus evolves within a specific individual. This requires an entirely different scale of data analysis, which is an order of magnitude greater than what I just described, because now you have millions of sequences per host. We're collaborating with folks at Stanford to develop a detailed resource for annotating mutations with clinical experimental data. We're studying the evolution of SARS-CoV-2 in companion animals, so horses, cats, dogs, raccoons, anything that we can find with veterinarians at Cornell. And we're also comparing the evolution of SARS-CoV-2 and other human coronaviruses with a team at Oxford and Glasgow. I would like to acknowledge my collaborators and a bunch of institutions that provide computing infrastructure for these large-scale efforts. And I thank you for staying on to listen to this talk. Hopefully you learned something, and I'd be happy to take any questions you have.